This is the Professor, CBMD PhD, and today is Wednesday, September 27, 2023. It is 7:04 and 1 second a.m. Eastern Standard Time, and today's research, MicroRNA activity. So let's hop to it here. Researchers developed first method to study microRNA activity in single cells. Date September 21st, 2023. Source: Stockholm University. This is a huge improvement over existing state-of-the-art methods that require millions of cells and will, for the first time, allow researchers to study microRNAs in complex tissues such as brains. MicroRNAs are small molecules that regulate gene activity by binding to and destroying RNAs produced by the genes. More than 60% of all human genes are estimated to be regulated by microRNAs. Therefore, it is not surprising that these small molecules are involved in many biological processes, including diseases such as cancer. And to discover the function of a microRNA, it is necessary to find out exactly which RNAs are targeted by it. While such methods exist, they require a lot of material, typically in order of billions of cells, to work. The scientific investigators and microbiologists have developed a new method to detect microRNA targets at the level of single cells. Such cells are each around 100 millimeter in diameter and weigh less than a billionth gram and comprise the basic building blocks of living organisms. And with their new sensitive method, the researchers can follow microRNA targeting of thousands of RNAs during biological processes such as the cell cycle or differentiation into red blood cells. The scientific investigators have discovered in these processes, the researchers find that microRNAs, surprisingly, perform quite different tasks in each cell. In the future, it will be possible to also apply this method to study microRNA targeting in whole tissues to find out exactly what is happening in each of the many cell types that comprise complex organs such as brains. The research team want to understand and ultimately make mathematical models of gene regulation at the level of a single cell. The new method is a huge leap towards making this possible. And this is the Professor of Space Technology, Advanced Physics, Mathematics, Computer Science, and Nature of Research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have an awesome day.